All right, hello everyone. My name is Ashbey Manning. I'm a senior chemical engineering major and I do research with Dr. Brittany Gibbons. Cancer, respiratory disease, and heart disease remain in the leading causes of death in the United States. Chemical engineers can help tackle these critical health challenges using innovations in drug delivery, such as nanoparticles. Nano and microparticles are small spheres made out of a material such as a polymer. A polymer is just a long chain of repeating molecules. Think string of pearls. Each one of those pearls is what we call a monomer. And as that polymer breaks down in the human body, that monomer is able to be excreted in a way that does not cause additional harm. Many polymers can be used for this method, but our lab focuses on polycaprolactone, or PCl. PCl has advantages over other biocompatible biodegradable polymers and that it has a long degradation time compared. This makes it a prime use for targeted and sustained drug delivery. That's why in our lab, when we synthesize these particles, we load them with active pharmaceutical ingredients, or APIs. APIs are the part of a drug that produces the effect. So when you have a headache and you take Motrin, the ibuprofen, which is the API, is what's getting rid of your headache. Now, in order to synthesize these particles, we use an oil and water emulsion. In our oil phase, we have the polymer, solvent, and sometimes a drug. And in the water phase, we have water and something called a surfactant, which is just an agent that allows us to form our emulsion. When we combine our oil and our water phases, like when you shake salad dressing, and then we stir them to evaporate the solvent off, we find that we make particles with API on the inside and polymer on the outside. In order to control the size of these particles, which is critical for applying it to a variety of healthcare problems, such as tumors versus lung disease, we control the amount of polymer or water that we use in our emulsion. Now, these particles are very small, so in order to see them, we have to use something called scanning electron microscopy, or SEM. SEM is just a fancy microscope that allows us to see things invisible to the naked eye. Here you can see our SEM images of two different formulations. When we use our SEM images, we were able to quantify the particle diameters. When we did that, we found that we were able to generate particles between 300 nanometers and 1.7 microns, which allows us to apply them to a variety of different applications. We also then tested our particles in human lung and endometrial cancer cell lines to ensure that the particles were making their way into human cells. Now, when we did this, we found that our particles were successfully being internalized in both cell lines within the first 4 hours. This makes it applicable for a variety of drug delivery applications. Now, based on these results, we have a few different future directions. So we were able to successfully generate our particles. We were able to get them in the size range that makes them applicable to a variety of different applications, but there are still future directions. So our target size range for tumoral drug delivery is 10 to 300 nanometers, and our smallest particles are currently right at 300 nanometers. So in order to look at how we can make these smaller, we're looking at things such as how polymer molecular weight, or how long that string of pearls is, might impact the sizing of the particles. We're also looking at initiatives to increase the yield and scale up of those small particles, as our cell studies are currently limited by the small amount of particles generated during this process. And with that, I open the floor for questions, and thank you for your time.